So now we're going to talk a little bit about active transport. So if we recall, active transport is different than passive transport in that it requires energy. And in active transport, remember that it requires energy because molecules are moving up their concentration gradient, or what we call against their concentration gradient. So molecules are moving up or against their concentration gradient. And since we're going against the concentration gradient and it requires energy, we also require a transport protein. So one type of active transport or transport protein is the Atpase pump. So the Atpase pump is one that uses energy from ATP, hence the name, to move ions across the membrane. So uses ATP to move ions across the membrane. And so this is a transport protein. It's going to bind the ion. There are a couple different types. There's the calcium ATPase pump, there's a sodium potassium ATPase pump, and we're going to take a closer look at that in just a minute as an example. But we have different types of ion pumps and ATPase pumps. So the ion is going to bind to the transport protein and also ATP. So it binds the iron and the ATP. And then the ATP is converted to ADP plus the phosphate group. The energy from this conversion, energy from removing that phosphate group, changes the shape of the carrier. And this forces the ion across the membrane. So this is important to create concentration gradients that are needed in the body. So if we want to make something go against its concentration gradient or up the concentration gradient, that's because we want a lot of a particular ion in a particular area. And this, for example, we have the calcium ATPase pump. And this calcium ATPase pump is going to actively pump calcium out of the muscle cells to end a contraction. So we send a message to our muscles saying that we want to contract those muscles, and then we do, but then we want that contraction to end. And so we need to get rid of the calcium in the muscle cell so that we can end the contraction. The other example I briefly mentioned already is the sodium potassium ATPase pump. And so in this situation, we're moving 3 sodium out of the cell and 2 potassium in the cell. And this is important for information to be sent along nerves. It's called an action potential. And this is important because we have to have these concentrations of sodium on one side and potassium on the other side. And then we need to change that concentration. So let's really quickly take a look at the sodium-potassium pump. I'm going to make sure that this is correct here. So we have our sodium-potassium pump, sodium-potassium exchange pump, or ATPase pump. Any of those works for this. And what we see here is that on the left-hand side, we have our sodium ions here. So you can see these sodium ions, and there are 3 of them, and they're going to attach to the sodium-potassium pump right here. There are 3 kind of indents there. They're the sites for connection for the ions, and it only is going to fit sodium ions. And so these 3 sodium ions are going to sit right in these seats, and it's always 3. And then over here we have the ATP. So the ATP is going to attach, the 3 sodium ions are going to attach, and when that happens, the ATP is then going to be broken apart into the ADP and the phosphate group. Once it's broken apart into the ADP and the phosphate group, then that is going to change the shape of the sodium-potassium pump. You can see here that this is what it looks like initially, and then over here we have changed the orientation. Now the sodium is moving out of the cell. So the sodium is moving out of the cell, and then at the same time we have potassium ions. They're labeled over here in the upper left-hand corner, but we have 2 potassium ions that are going to now sit in their little sites, their little seats, and they're going to attach. And then what happens is our ADP and our phosphate group are going to be fallen away. We have another ATP here. This ATP is going to attach, and then it's going to change the shape of the sodium-potassium pump, and then the potassium is going to leave the pump and go inside of the cell. So we have our 2 potassium here. And then we are back to where we started, all the way over here. So we have our ATP is attached, our 3 sodium ions are going to go in their little seats, and then it's going to go over and over and over again. So this is a really good example, a very common example of a transport protein in active transport, is our sodium-potassium pump. Now this is what's called primary active transport. We also have something called secondary active transport. And in secondary active transport, what we see is that we are going to use the energy of a concentration gradient to transport a molecule using a different transport protein. So it uses the energy of a concentration gradient to transport a molecule. So this is going to be a situation where we allow one molecule to move down its concentration gradient and then use that energy of that concentration gradient to move a different molecule up its concentration gradient. So use one molecule going down the concentration gradient while the other goes up the concentration gradient. And that's because it's using the energy from the first concentration gradient. So for example, we have what's called a sodium glucose symporter. Symporter, meaning working together. And so our sodium glucose symporter is where we have our sodium is low inside the cell due to what we just talked about, that sodium-potassium pump. So our sodium is low inside the cell because of our sodium-potassium pump that's been pumping sodium out of the cell. But sodium wants to move inside the cell. So sodium wants to move inside the cell. And this is because it wants to move down its concentration gradient. So then we allow sodium into the cell, moving down its concentration gradient. So this is allowed. It's moving down its concentration gradient. And then when the sodium is moving into the cell, it brings glucose in also. So as the sodium is moving down its concentration gradient, brings glucose with it. And then glucose, we're moving up the concentration gradient or against the concentration gradient. Because we already have high amounts of glucose in the cell, but we want to keep bringing that in and keep bringing that in. So even if glucose is high in the cell, it's going to continuously do that. And even if it's low in the cell, it's going to be doing that because it's going to be bringing it in with it each time. So this is our active transport. We had our primary active transport, which was our sodium-potassium pump example, and our secondary active transport, which is the sodium-glucose symporter. So where we're using the concentration gradient of 1, the energy of that, to then have the protein, the transport protein, move something against its concentration gradient. So the last type of membrane transport that we need to talk about is vesicular transport, or bulk transport. So the first type of vesicular transport, or bulk transport, that we're going to talk about is endocytosis. And endocytosis is where we're going to be bringing molecules or particles into the cell. So bring molecules or particles into the cell. So the way that this works is we're going to actually be using the plasma membrane of the cell to bring something into the cell. So endo as like into, endo is to bring something into, and then cytosis. Cyto is related to cell. So it's bringing something into the cell. So the way this works is that the membrane, the plasma membrane, is going to surround a molecule or surround an object or surround some particles, and then it forms a vesicle. And then that vesicle is going to pinch off from the plasma membrane and then come into the cell. So the membrane is going to surround a molecule or an object, and then it forms a vesicle, because all of these things are made up of plasma membrane, and then it contains the object in the cell. So if we take a look at this, we have our plasma membrane. All right, so our plasma membrane here. I don't know why we have it curved here, but there's our plasma membrane. And then let's say we have something come along, like a bacteria. We want to grab onto it, bring it into the cell, and gobble it up. So we have a bacterium, and the cell is going to grab onto that and bring it in via vesicular transport. So something really large, like a bacterium. If you can imagine, if this is our plasma membrane here, our transport proteins would be something like that size. Let's do it in different colors. So something like this size. So if we're looking at a bacterium and we're looking at the transport protein, we're certainly not going to be grabbing onto a bacterium and bringing it into the cell to break it down through something that's that size. It's much, much too large. And this is why we would use something like vesicular transport or bulk transport. And so then what happens is that we end up having these little pseudopods of the plasma membrane, which is where our plasma membrane is going to start to change shape. And so our bilayer is going to start to come out just like so. So this is no longer like this. And in fact, remember, it's a double plasma membrane, so the plasma membrane, both sides, are going to be starting to make these little pseudopods, which are called pseudopods, which mean fake feet. So our plasma membrane is going to start to create these kind of like little arms, or little fake feet, that are going to start grabbing onto things. So they're going to go and they're going to continue around, and then they're going to meet on the other side of this object. So then when this happens, what we end up seeing is something like this. So we have merged on the other side of this object. This is our bilayer, our plasma membrane. And then we have something that looks like this, with our bacterium in the inside. So you can see if this continues going up and they glob onto each other on the other side, if you follow, this is our plasma membrane, then it's going to go up, and then it's going to have this big kind of globule on the inside. So then what happens is the plasma membrane, as it's fluid and can move around, this is going to then merge and just be the single plasma membrane on the outside of the cell here, meaning the bilayer, the regular bilayer, and then this here is going to pinch off. And so then we're going to have our bilayer here, and then we have our vesicle that has now entered the cell. Now this is an example of endocytosis, where we're bringing molecules or particles into the cell. Now we have three different types of endocytosis. The first type, and the one that I just illustrated, is phagocytosis. So phago is related to eating. This is called cellular eating. And when we're talking about something like cellular eating, we're engulfing large particles like bacteria. So that would be phagocytosis, is what we just illustrated here. Now another example is pinocytosis. And pinocytosis is actually cellular drinking. And in this case, what we're doing is we're going through the same kind of process, but instead of a bacteria being in here, this is just going to be a bunch of extracellular fluid inside the vesicle. So we won't have the bacterium, but we'll have a bunch of fluid from the outside. And the reason that this is grabbed onto is because then we have all of these different particles that are in the extracellular fluid. So it could be different ions, it could be huge molecules or bigger molecules, like glucose and other larger molecules that we want to bring in bulk. So remember, this is our bulk transport. So pinocytosis is cellular drinking. We're not grabbing onto a single object or a single item like a bacterium, but we're just kind of grabbing onto a bunch of the fluid that's going to be containing a lot of the stuff on the outside of the cell. Now the third type of endocytosis that we're going to talk about is receptor-mediated endocytosis. So receptor-mediated endocytosis is, as its name implies. In this case, what we have is, if I am to, let's say we combine this here, and then let's say we have little receptors. So we have little tiny receptors on the plasma membrane. And if we have these little tiny receptors, then when the molecule, whatever the molecule happens to be, binds to these receptors, then what happens is we start what's called invagination. So the invagination is what we're talking about when we're talking about endocytosis. Invagination is the plasma membrane starting to dip down, create these pseudopods. So here our plasma membrane is dipping down here and then creating these pseudopods. And in this case, we would see a similar thing. This is receptor-mediated endocytosis. We're using receptors. The molecule is attaching to the receptors, and that initiates endocytosis, bringing it into the cell. And so then what we see is that this is going to start coming up. So the plasma membrane starts to come up, and then we're going to see that combine around that. And so then actually, rather than drawing this all over again, this is actually kind of moving down as the plasma membrane is moving around it. So this would be our receptor-mediated endocytosis. So when we're talking about endocytosis is one type of vesicular transport. It's bringing something into the cell. And then we have the three types of endocytosis. Now the next type of vesicular transport that we are going to talk about is exocytosis. And exocytosis is when we release molecules to the outside of the cell. So in this case, I usually think of it as exiting the cell. Exocytosis is exiting the cell. And so in this case, we have our vesicles are going to fuse with the plasma membrane and release the contents to the outside. And this is what we saw and what we've already talked about with our rough endoplasmic reticulum, making the vesicles and moving them to the Golgi apparatus or the Golgi body. And then those having vesicles, it packages and sorts and modifies things and packages them up in vesicles. And then what we have are these vesicles that are filled with molecules that we have made, maybe different proteins, glycoproteins, whatever the case may be. And then these are going to move to the plasma membrane. It's going to merge with the plasma membrane. All of these molecules are going to become part of the plasma membrane here. And then what we see happening is that, let's open up here, we end up opening up this vesicle here. to the exterior. So then all of the contents, so once we've merged this thing, all of the contents then are able to leave the cell because we've had this merging. So this is a step-by-step process here. And this is exocytosis, so exiting the cell. So our vesicles are going to fuse with the plasma membrane. And then release the contents outside. So some examples of this are secretion of hormones. So a lot of times we make our hormones in the cell. We can make our lipid hormones. We can also make our protein hormones. And we secrete those so that they can go to where they need to go. Or release of waste products. So that is exocytosis. So we had our vesicular transport, that sums up our vesicular transport, which is endocytosis and exocytosis. And endocytosis is further broken down into phagocytosis, which is cellular eating, pinocytosis, which is cellular drinking, and then our receptor-mediated endocytosis, which utilizes receptors to then initiate endocytosis. So the last thing we're going to talk about in our cells here is the membrane potential. So the membrane potential is the charge of a cell. And what that means is that there is a difference between the charge inside a cell and outside of the cell. So if we have our plasma membrane here, we have the inside of the cell, just down here, inside, and the outside of the cell. And there are different charges in the inside of the cell and the outside of the cell. And this is due to the different ions. So let's see, different ions inside cell versus outside of the cell give different charges. Inside versus outside. So we have different concentrations of ions. So different ions, but then also different concentrations of ions in the inside of the cell versus in the outside of the cell. And so what this means, actually, is that we tend to have a lot of negative ions right along the inside of the cell. So we have a lot of negative ions right up against the inside of the plasma membrane and the inside of the cell here. And then on the other side, on the outside, we happen to have a lot of positively charged ions over here. And so we have this difference between the negative inside and the positive outside. And this provides a voltage. So a change in charge between the inside and the outside gives us a voltage. And so what is on the inside of the cell here, these right here, the negative charge, we have a lot of negatively charged amino acids, for example. Amino acids that would be inside of the cell. A lot of the amino acids are negatively charged. We also do, however, have potassium inside of the cell, and potassium is positively charged. But it tends to be floating around in the inside of the cell, and it tends to be around these negatively charged amino acids, and the negatively charged amino acids tend to kind of pull up next to the plasma membrane. On the opposite side, these positively charged ions are mostly sodium ions. So we have a lot of positively charged sodium ions along the plasma membrane on the other side. We do, however, have some negative ions, so we have some negative chloride ions, which we have over here as well, different negative chloride ions as well. We do also have some positive potassium over here, but again, not in any quantity that's going to be specifically related to this charge. We tend to have higher amounts of potassium inside of the cell and higher amounts of sodium outside of the cell. So So we'll have a little bit of sodium over here, but not too much. And so then the net difference here is that we have more of a negative charge on the inside and more of a positive charge on the outside. And so this is what's called a membrane potential. This charge difference between the inside of the cell and the outside of the cell is this membrane potential. This is the resting membrane potential. I'll probably add that up here. So the resting membrane potential. So what that means then is that the membrane potential of most cells is negative. And that's because we're talking about the inside of the cell, which is negative. Potential. All right, and that's because it's more negative inside compared to more positive outside. And so we know that this is the case because if we think about our sodium-potassium pump, right, our sodium-potassium pump is in here. Remember, it is going to take sodium, right? So our sodium here, we have our 3 sodium that are going to be attached to the sodium-potassium pump. Remember, 1, 2, 3, they sit in their little seats there. And then we have ATP is going to be attached. Then we have a change in shape when the ATP is then changed to a DP and the phosphate group, an organic phosphate. So this is ADP, ATP. And then that releases our sodium ions to this side of the cell. So we just put 3 positives on the outside of the cell. Now, if we remember the sodium-potassium pump, what that means then is at this point we have 2 potassium that are going to attach to the sodium-potassium pump. And then when this happens, we have a change in conformation. We have an ATP that's going to attach to the sodium-potassium pump, and that's going to change. And we have those 2 potassium are going to then move inside of the cell. Now what we've done here, we have this sodium-potassium pump. We have lots and lots of sodium-potassium pumps doing this all throughout the cell. But if you'll look, what we've done is we've put 3 positives on the outside of the cell and only 2 positives on the inside of the cell. And so that gives us this slightly negative charge on the inside because we don't have as much positive on the inside as we have on the outside. And so we have more negativity on the inside, more positive on the outside. Now, some cells can change their membrane potential. And so that's why I also mentioned that this is a resting membrane potential. So some cells, like nerve cells and muscle cells, can change their membrane potential. And they do this in response to signals. So we send a signal. Let's say we send a message from the brain to our leg muscle because we want to move it. When we send that message to our muscle cell, the muscle cell is going to change its membrane potential. And it does this by letting ions in or out. So for example, we can open sodium channels. And if we open sodium channels, that's going to allow sodium to come into the cell. So sodium comes in cell. And then that's going to increase the positive charge in the inside of the cell. We can also open potassium channels. And that is going to allow the potassium to leave the cell because there's more potassium inside the cell than outside the cell, so it's going to go down its concentration gradient. And that'll increase the negative charge inside of there, so it'll make it more negative because that positive potassium will be leaving. We could also open chlorine channels. And if we remember, our chlorine is higher outside the cell than inside of the cell. So then the chlorine will come in, and that will increase the negative charge in the inside of the cell. And so we can we can manipulate the membrane potential. We can make it more positive, we can make it more negative, depending on the specific channels that we have open and how active our ATP pumps are, our ATPase pumps, the sodium potassium pumps. So we do this in response to these signals. And these signals are going to bind to receptors that are going to open these ion channels. So these signals are going to bind to receptors that open channels. And so then we can open these channels, and then it's going to change the membrane potential. So that kind of summarizes our cells section, finished up our transport with our active transport information requiring energy. We talked about the ATPase pump, specifically the sodium potassium pump, which is a really important one to understand. We also talked about secondary active transport, so talking about the sodium glucose symporter. And then we talked about our vesicular transport. We talked about endocytosis, bringing something into the cell. We talked about exocytosis, having something leave the cell. And then remember, there are 3 different types of endocytosis. There's phagocytosis, which is cellular eating, which is large particles like bacteria. There's penocytosis, which is cellular drinking, bringing in kind of the fluid that's outside of the cell, the extracellular fluid. And then receptor-mediated endocytosis. So when we have receptors that attach to the molecules, and it initiates bringing that vesicle into the cell. And then lastly, we talked about the membrane potential. So we talked about how there's a resting membrane potential, which means that the cell is more negative on the inside than it is on the outside. But then our nerve cells and our muscle cells can change that membrane potential. And that's in response to signals that are sent, and we can manipulate the channels and change the direction that our sodiums are not sodiums, but our ions are flowing to adjust the membrane potential. We can make it more positive, then we can make it more negative, depending on what we open and what we close and when we do that.